This is an eukaryotic cell which has a true well-defined nucleus. Inside the nucleus, this is the DNA. From DNA, messenger RNA is formed. The polarity of the messenger RNA is 5' to 3'. The messenger RNA which is formed inside the nucleus is called primary transcript or heterogeneous nuclear RNA. This primary transcript or hnRNA is not mature, it is not functional, it is not active. It cannot translate polypeptides. It cannot start protein synthesis because it is immature. To become mature, the primary transcript, the newly formed mRNA, is processed. It undergoes certain modifications, processings inside the nucleus after transcription is complete. After the processings, after the modifications, this will be fully mature, functional. Then it will be transported to the cytoplasm for protein synthesis. So inside the nucleus of eukaryotic cell, the primary transcript or hnRNA undergoes processing to become mature. There are three processings. There are three types of modifications. This is the hnRNA or immature non-functional primary transcript. It is not functional, it is not mature because it consists of exons and introns. Exons are coding sequences, introns are non-coding sequences. So the newly formed messenger RNA in the nucleus is not mature, it is not functional because it consists of exons and introns. Now it undergoes processing inside the nucleus. First of all, it will undergo splicing. Splicing means the introns are removed and the exons are joined in a definite order. Splicing. Splicing means the introns are removed and the exons are joined in a specific order. After splicing, capping. Capping means to the 5' end of the primary transcript or newly formed messenger RNA, a cap is added. This is called 5' cap. The 5' cap is actually a compound. This compound is 7-methyl-guanosine triphosphate. 7-methyl-guanosine triphosphate is added to the 5' end of the immature messenger RNA. This is called 5' cap and the process is called capping. Now tailing. After capping, the primary transcript will undergo tailing. Tailing means a tail is added to the 3' end of the primary transcript. The tail is called poly-A tail. The tail is added to the 3' end of the hnRNA or primary transcript. The tail is called poly-A tail. The tail consists of 200 to 300 adenylate residues. That means 200 to 300 adenylate residues are added to the 3' end of the newly formed messenger RNA. Adenylate means adenosine monophosphate. Adenine plus nitrogen base plus phosphate. Adenosine monophosphate. Adenylate means a nucleotide which consists of adenine, one phosphate and one nitrogen base. This is called adenylate or AMP. So 200 to 300 adenylate residues or adenosine monophosphates are added in a template independent fashion. Template independent fashion means without using template, without depending on the sequence of the template, without using any template, 200 to 300 adenylate residues are added to the 3' end. The formation of poly A tail occurs without using any template. It does not depend on the sequence of any template. So this occurs in template independent fashion. In this way, the newly formed messenger RNA becomes mature functional after it undergoes splicing, capping and tailing. It becomes processed. This is the fully functional, mature and processed mRNA ready for translation. Now it is ready for translation. Now during splicing, to remove the introns and to join the exons, some proteins associate, which are called small nuclear RNAs with proteins. These are small nuclear RNAs, and some proteins associate, which together help in the process of splicing. Splicing means removal of introns. So because of splicing, exons are joined. So in this process, some proteins and small nuclear RNAs associate and bind to the messenger RNA. Some proteins and small nuclear RNAs associate and they bind to the messenger RNA to help in the process of splicing, to remove the introns and to join the exons. Small nuclear RNAs, these are the RNAs which associate with proteins and bind to the primary transcript to remove the introns and join the exons in a specific order.